We all know the usual suspects when it comes to atherosclerosis, high cholesterol, high blood pressure.
Right, the big ones.
But have you ever wondered about those cardiovascular events, the heart attacks, the strokes that happen even when those classic risk factors seem, well, under control?
It happens more often than you might think.
Yeah, it turns out focusing only on those well-known causes might mean we're missing a pretty significant piece of the puzzle.
That's absolutely right. And today we're going to take a deep dive into some of those, let's say, less commonly considered contributors to atherosclerosis.
Okay.
We're drawing heavily from the 10th edition of Rutherford's Vascular Surgery and Endovascular Therapy. So, solid source material.
Excellent. We're going beyond the everyday understanding then.
Exactly. We want to explore the fascinating complexities that are really at play here.
Okay, so forget just LDL and HDL for a minute.
Well, don't forget them. They're still crucial.
Huh, okay, fair point. But let's look beyond just them. We're going to unpack some surprising things about the atherosclerotic lesion itself, right?
Uh-huh. And explore how hyperlipidemia involves more than just those two main types of cholesterol.
And then get into diet, but not just fat content,
Right, things like the gut microbiome.
which is huge right now. And infections, past infections.
Yeah, the potential role of past infections. And even environmental toxins, things we breathe, things we absorb.
Wow, okay. So our mission today really is to connect some of these maybe unexpected dots and give you, the listener, a much more complete picture of atherosclerosis.
Precisely. We want to equip you with insights into these, well, less obvious factors.
Okay, let's unpack this. Where better to start than the core of the issue, the atherosclerotic lesion itself, the plaque.
Makes sense.
So the traditional story is pretty familiar, isn't it? Damage to the artery lining kicks things off.
Yep, endothelial injury, that's the trigger.
Then inflammatory cells, monocytes, they arrive on the scene.
They migrate in, and then they transform into macrophages.
Right, and they start gobbling up oxidized LDL cholesterol, becoming these fat-filled foam cells.
Exactly. And then, to sort of wall it off, vascular smooth muscle cells, or VSMCs, migrate there too, forming a fibrous cap over the whole thing.
Seems straightforward enough. A patch job,
That's
basically.
the foundational understanding, yeah. And it's accurate as far as it goes. But recent findings, they reveal that the cells within the lesion, the endothelial cells, the VSMCs, the macrophages, they're much more adaptable plastic than we originally thought.
Adaptable how? Like their roles aren't set in stone.
Exactly. That's the key. Their jobs aren't fixed.
Okay, here's where it gets really interesting then. It sounds like under these atherogenic conditions, these artery clogging conditions, the cells can actually change their identity.
That's what the evidence points to. Endothelial cells might transform into cells that look and act more like fibroblasts, which are usually involved in scarring. And maybe even more surprisingly, those VSMCs, they can shift into an inflammatory macrophage-like state.
Whoa! So the muscle cells start acting like the inflammatory cells. It's like they're shape-shifting inside the artery wall because of the environment.
That's a good way to put it, shape-shifting. And what's fascinating from a research perspective is the challenge this creates.
How so?
Well, when you're looking at a plaque in a living system, in vivo, it becomes really difficult to tell apart foam cells that started as macrophages versus those that came from these transformed VSMCs.
Why? Don't they look different?
They start expressing similar markers on their surface. Things like CD68 and MAC2, which we used to think were mainly on macrophages, can show up on these altered VSMCs.
Huh.
And at the same time, the VSMCs might reduce their own typical markers, like alpha-smooth muscle cell actin. So it really blurs the lines of, cellular identity within that plaque.
Okay, so these transformed VSMCs acting like foam cells.
Sure.
What kind of trouble are they causing then? Are they just filling up space?
Oh, they can contribute to the lesion's progression in pretty significant ways. We see increased rates of cell death, apoptosis in these altered VSMCs.
That doesn't sound good for plaque stability.
It's not. And critically, their ability to help remove cholesterol, that process called reverse cholesterol transport, seems to get impaired. They become dysfunctional in that key protective role.
So wait a minute. The very cells, the VSMCs, that are supposed to be stabilizing the plaque by building that fibrous cap
Yeah.
are also potentially contributing to its instability by turning into these problematic foam cells. It's like the construction crew is also secretly, undermining the foundations.
That's a really apt analogy. It perfectly underscores this dual role of VSMCs in atherosclerosis.
Good guys and bad guys, all in one cell type.
Kind of. They are vital for forming that protective cap, which gives the plaque strength, but they can also add to the dangerous foam cell population that makes the plaque vulnerable to rupture.
So understanding this,
Hmm.
this plasticity, this shape shifting, it changes how we think about treating plaques.
It really does. It suggests our understanding of plaque vulnerability needs to evolve. Maybe the focus shouldn't just be on shrinking the plaque, but also on finding ways to stabilize it, perhaps by influencing these VSMC transitions.
And it gets even more nuanced, doesn't it? You mentioned it's not like all VSMCs are identical.
Exactly right. VSMCs aren't just one single type of cell. They can actually originate from different places during our development.
Like where?
Some might come from precursor cells in the bone marrow, others from the outer layer of the artery, the adventitia, or from the main middle layer, the media itself.
And did that matter? Where they come from?
It seems to. Cells from these different origins might behave differently and have varying effects once they're inside that atherosclerotic lesion.
Wow. This sounds like a whole new level of complexity just within the plaque itself. So what does this all mean for us? Why should we, as listeners, care about where these smooth muscle cells originally came from?
Well, the implication is, if we can truly understand these different VSMC populations and what each specific type is doing, which ones are helpful, which ones are harmful,
Yeah.
it could potentially lead to highly targeted therapies down the road. Imagine being able to selectively boost the protective functions of some VSMCs while, limiting the damaging contributions of others.
Okay, that makes sense. Targeted intervention based on cell origin. That's the potential long-term payoff.
That's the hope driving a lot of this research.
All right, let's pivot a bit. Moving from the intricate world inside the lesion to something we hear about all the time, hyperlipidemia, high cholesterol.
Okay.
We all know LDL, the bad cholesterol, and HDL, the good cholesterol, are the main targets, right?
Yeah.
That's what our doctors usually focus on.
They are definitely primary targets, yes. Critically important.
But you mentioned earlier there's more to the lipid story than just these two.
Yes, absolutely. While LDL and HDL management is fundamental, there are additional lipid-related variables, other fat-like molecules in the blood, that have really emerged as potential independent risk factors.
And these contribute to what you called residual risk.
Exactly. That's the risk that hangs around even when someone's LDL cholesterol looks great on paper. Often thanks to statin therapy, there's still something else going on.
Okay, so what's one of these other players?
A major one gaining a lot of attention is lipoprotein, usually written as LPA.
LPA. Okay, tell us about this one. What is it?
It's a really fascinating molecule structurally. Think of it as basically an LDL particle, the bad cholesterol particle, but it has another protein tacked onto it. This extra protein is called apolipoprotein, or just APOF for short.
So it's like LDL plus?
Kind of, yeah. And both parts can cause trouble. The LDL-like part can get oxidized, just like regular LDL, promoting plaque formation and inflammation. Standard stuff.
Okay.
But the APO-A part is also significant. It has a structure that's weirdly similar to another protein called plasminogen.
Plasminogen. That rings a bell. Isn't that involved in breaking down blood clots?
Exactly. Crucial for clot busting. So because APO looks like plasminogen, it can actually interfere with that clot breakdown process.
Get out. So it might have prothrombotic effects. Makes clots more likely or harder to break down.
That's the concern. So LPA could be a double whammy, contributing to plaque buildup and making clots stickier.
Wow. That sounds like a significant independent factor then.
It really seems to be. And what's clinically important is that studies show LPA levels correlate with both the initial development of cardiovascular problems and also their recurrence, like having a second heart attack.
And this is separate from LDL and HDL levels.
The association appears to be independent of LDL and HDL cholesterol levels, which is key. It suggests that someone could have perfectly normal cholesterol by standard measures, but still carry the significant hidden risk because of high LPA.
That's a pretty big deal then if it's acting on its own track. What can actually be done about high LPA? Is it treatable?
Well, current approaches, things like statin medications and niacin, they might offer some modest reductions in LPA, but often not dramatic.
Okay.
Apheresis is another option. That's a more intensive procedure, kind of like dialysis, where blood is filtered to remove specific things. It can remove both LDL and LPA particles.
Right, I've heard of that for severe cholesterol issues.
And interestingly, there's some evidence suggesting that LPA-specific apheresis, when added to the best medical therapy, might actually slow down the progression of coronary artery plaque buildup and reduce major cardiovascular events.
That sounds promising, but apheresis, that sounds pretty involved. Probably not something everyone can easily access or undergo regularly.
It's definitely more complex and resource intensive, yes. But what's really exciting on the horizon is the development of new drugs, specifically subcutaneous antisense oligonucleotides. These are basically designer molecules, injectable, that are engineered to specifically reduce the body's production of that lipoprotein.
So they stop LPA from being made in the first place.
Essentially, yes. By reducing Alpo, they lower the overall LPA levels, and these look very promising. They might offer a much more practical, widely applicable way to tackle high LPA compared to apheresis.
Okay, so tying this back, if conventional statin therapy tackles LDL effectively, but might only address part of someone's overall cardiac risk,
Yeah.
could considering LPA, testing for it, treating it, be a crucial step in tackling that leftover residual risk?
That's the key insight here. That's exactly the thinking. By looking beyond just LDL and HDL and bringing LPA into the picture, we might be able to identify and help individuals who are still at significant risk despite having seemingly well-managed cholesterol by the standard numbers.
Fascinating. Okay, let's shift gears again. We've talked lipids in the blood. Now let's talk about what we put in our bodies. Diet.
Always a big topic.
We all know the general advice, link between high-fat, high-cholesterol diets and heart disease. But you mentioned earlier there might be more to the dietary story than just the amount of cholesterol we eat.
Absolutely. The focus is definitely broadening. It's not just about fat and cholesterol anymore. We're looking more at other dietary components and how they interact with our bodies, especially our gut microbes.
The microbiome again.
Yes, and this leads us to something called trimethylamine N oxide, or TMAO.
TMAO. Okay.
This molecule has been getting a lot of attention recently for its protherogenic effects, meaning it seems to promote atherosclerosis.
TMAO. Okay, where does that come from? It's not directly in our food, is it?
No, it's not. It's actually produced through this fascinating multi-step process that heavily involves our gut bacteria.
Okay, walk us through it.
Certain compounds in our diet are the starting materials, primarily L-carnitine,
Which is found in?
abundant in red meat, and also phosphatidylcholine, or PC,
And PC is in?
found in many animal products, things like egg yolks, liver, dairy, fish, too.
Okay, so red meat, eggs, dairy.
When we eat foods rich in carnitine, or PC, specific bacteria living in our gut metabolize these compounds and convert them into something called trimethylamine, or TMA.
So the bacteria make TMA first.
Correct. This TMA then gets absorbed from the gut into our bloodstream, travels to the liver, and in the liver, a family of enzymes called flavin monooxygenases, FMOs for short, especially one called FMO3, takes that TMA and oxidizes it, converting it into TMAO.
Wow. So it's diet, gut bacteria, liver enzyme, last TMAO in the blood.
You got it. It really highlights how our gut microbiome isn't just passively digesting food, it's actively metabolizing dietary components into molecules that can directly impact our artery health. It makes cardiovascular risk quite personalized, depending on your diet and your specific gut bugs.
So our gut bacteria are playing this absolutely pivotal role in producing this potentially harmful molecule.
Precisely. And here's another layer of complexity, or maybe convergence.
Go on.
Remember those FMO enzymes in the liver, particularly FMO3?
Yeah, the ones that make TMAO.
Well, they also play a role in detoxifying various drugs and, importantly, environmental toxins, including those persistent organic pollutants, the POPs we'll probably talk about later.
Wait, really the same enzyme?
The very same, FMO3. So this raises the possibility of a kind of synergistic effect. Meaning your diet, by providing the TMA precursors, and your exposure to certain environmental toxins, which might affect FMO3 activity, could potentially work together to exacerbate atherosclerosis risk via this TMA over pathway.
Interesting. Okay, so if TMAO is something we generally want less of, Can we do anything through diet or other means to lower its production?
Well, since its production depends so heavily on gut bacteria, you can actually suppress TMAO production quite effectively with antibiotics.
Makes sense. Kill the bacteria, stop the TMA production, though antibiotics long-term isn't ideal.
Not usually, no, but it demonstrates the principle.
Well,
More relevantly, the amount of animal products in your diet significantly influences the composition of your gut microbiome.
how so?
Basically, what you eat feeds certain bacteria more than others. So, a diet high in red meat and other animal products tends to promote the growth of bacteria that are good at making TMA.
Ah, okay.
Consequently, people who eat a lot of animal products tend to produce more TMAO. Vegetarians and vegans, for example, generally have much lower TMAO levels compared to omnivores.
And can this change quickly?
It seems so. Even short-term dietary changes, like switching to a plant-based diet for a period, can lead to noticeable shifts in your gut microbial populations and lower TMAO production.
It sounds like there's maybe a bit of a reinforcing cycle happening here then.
Exactly. It seems that consuming animal products not only provides the fuel for TMAO production, but also helps cultivate a gut environment where the TMAO-producing bacteria thrive. It's a positive feedback loop, potentially.
Okay, and the evidence linking TMAO itself to heart disease. What does that actually look like in human studies? Is it a strong link?
It appears quite strong, yes. Numerous studies in humans have shown a pretty consistent association between higher blood TMAO levels and an increased risk of various cardiovascular problems.
Like what specifically?
Peripheral artery disease, PAD, heart attacks, myocardial infarction, major adverse cardiovascular events, MACE, coronary artery disease, CAD, and even overall mortality, dying sooner from any cause.
Wow, that's a broad range.
Yes, and studies that have looked specifically at diets supplemented with TMAO precursors, like choline related to PC or L-carnitine, also generally suggest higher risks of atherosclerotic diseases and death specifically from cardiovascular causes.
But you mentioned earlier some seemingly contradictory evidence, particularly around L-carnitine supplements and maybe different types of meat.
Yes, the picture isn't entirely, shall we say, black and white. It needs more nuance. For example, some research, particularly in animal models, suggests that L-carnitine supplementation, while it does increase TMAO levels, might paradoxically have some protective effects against atherosclerosis in those models. And there's even some suggestion it could be beneficial for humans after a heart attack. Also, some studies looking at meat consumption have hinted that the increased cardiovascular risk might be more strongly linked to processed meats rather than fresh or frozen unprocessed meats.
Okay, that's a bit of a head scratcher then.
Yeah.
Higher TMAO, but maybe protective effects from the source?
Right.
Or processed versus unprocessed?
Yeah.
How do we interpret these different findings?
It's tricky. One issue is that some of these studies might not have fully accounted for the impact of non-meat animal products like dairy and eggs on TMAO levels. They might be confounding the picture.
Right.
So more research is definitely needed. What we really need are well-designed human intervention trials studies where you actively manipulate TMAO levels, maybe through diet or other means, and then look directly at whether that changes the risk of major cardiovascular events like heart attacks and strokes. That's the kind of evidence that would really nail down TMAO as a modifiable risk factor.
Makes sense. Got to prove cause and effect. Now, you've really highlighted the gut microbiome's role in this TMAO story, but it sounds like this, vast community of microorganisms living inside us might have even broader implications for atherosclerosis beyond just making TMAO.
Oh, absolutely. The interaction between our diet, our gut microbiome, and atherosclerotic disease is, frankly, one of the hottest areas of research right now. It's incredibly active.
So beyond TMAO production,
Yep.
what else is the microbiome doing that matters here?
Well, fundamentally, the microbiome plays a huge role in how we absorb and metabolize a whole range of nutrients from our diet, not just the TMAO precursors. It affects calorie extraction, vitamin synthesis, processing of fibers, all sorts of things.
And are there specific types or maybe imbalances in our gut bacteria that seem to be linked more closely to heart health issues?
It's still relatively early days in terms of mapping out the, literally trillions of bacteria in the human gut and understanding all their variations between people. But yes, certain patterns do seem to emerge more frequently in individuals who have atherosclerotic disease or its risk factors compared to healthy individuals.
Like what kind of patterns?
One common finding is a lower overall diversity of gut bacteria, like having fewer different species in the ecosystem.
Less diversity is bad.
Generally seems to be less resilient, yeah. Another pattern often seen, especially in people who are obese or have type 2 diabetes, Both major risk factors for heart disease is a shift in the balance of the major groups of bacteria. Specifically, a higher proportion of bacteria belonging to a large group called firmicutes relative to another large group called bacteroides.
Firmicutes versus bacteroidates, okay?
You can almost think of your gut like a diverse ecosystem, a garden. In some people, particularly those with weight issues or diabetes, certain types of bacteria, like many firmicutes, become more dominant. It's like a particular type of weed starts taking over the garden.
And these weeds, the firmicutes, what do they do differently?
Well, functionally, individuals who have this higher firmicutes to bacteroidates ratio seem to be more efficient at extracting calories from the same amount of food.
More efficient so they get more energy out of their food.
Exactly, which could contribute to weight gain. This imbalance is also associated with other metabolic changes. For instance, decreased production of a beneficial short-chain fatty acid called butyrate,
Butyrate.
which is important for gut health, and an increased production of another fatty acid called acetate. These shifts can contribute to low-grade systemic inflammation, reduced insulin sensitivity, making diabetes more likely, and the development or worsening of obesity.
Wow. So it's really not just about the calories we eat, but this incredibly complex interplay with our internal ecosystem and what those microbes are producing from our food that can ripple out to affect our wider health.
Precisely. It's a dynamic interplay. And the relationship seems to be bidirectional.
What do you mean bidirectional?
Well, we know that dietary changes and weight loss can shift the microbiome back towards a potentially more healthy profile, maybe lowering that firmicutes to bacteroidates ratio.
So we can influence the bugs.
Yes. And conversely, altering the microbiome directly, for example, through interventions like fecal microbiota transplants, mostly studied in animals, but with some intriguing human studies too, has shown some promise in actually promoting weight loss and improving insulin sensitivity in the recipient.
Fecal transplants. That still sounds pretty experimental for heart disease.
Oh, I thought definitely experimental in this context, yes. But it illustrates the principle that the microbiome itself can influence metabolic health. We need much larger, longer-term studies looking at actual clinical endpoints like heart attacks before we'd consider anything like that for atherosclerosis prevention or treatment.
Right. But it certainly opens up a whole new avenue for potential future interventions, doesn't it?
Yeah.
Thinking about therapies targeting the gut microbiome.
It absolutely does. The potential for microbiome-based strategies, whether through diet, prebiotics, probiotics, maybe even more targeted approaches in the future to help prevent or treat atherosclerosis, is a really compelling area of ongoing research.
Okay, let's shift focus again. We've done lipids, diet, microbiome. Now let's turn our attention to another area that, well, it's been a subject of debate for a long time. Infection.
Ah, yes, the infection hypothesis.
I definitely recall hearing years ago about potential links between certain infections like, I don't know, bacteria or viruses and heart disease. But then it seemed to become less of a focus, maybe.
You're right. The idea that infectious agents might contribute somehow to atherosclerosis has been around for decades. And yes, maybe some of the initial enthusiasm tempered a bit after some large antibiotic trials didn't quite yield the clear-cut positive results people were hoping for.
Right.
But that doesn't mean the idea is dead. The potential associations and even potential causal links between certain pathogens and the development of atherosclerosis are still very much under active investigation.
Okay, so what are some of the ways these bacteria or viruses might actually contribute to the process of plaque development? How could an infection lead to clogged arteries?
Several mechanisms have been proposed over the years. One idea is that the infection triggers inflammation, not just locally, but systemically, and that inflammation spills over into the artery walls.
Makes sense.
Another is that infections might cause the upregulation of adhesion molecules, sticky bits on the surface of the endothelial cells lining our blood vessels.
Making it easier for inflammatory cells to grab on and get in.
Exactly. Making it easier for monocytes and other cells to stick and then migrate into the artery wall, kicking off that plaque formation process. Direct damage to the endothelium by the pathogen itself is also a possibility.
Okay.
And interestingly, many pathogens seem able to stimulate the formation of those foam cells we talked about earlier, partly through receptors on our immune cells called toll-like receptors, or TLRs. Think of these TLRs as kind of like security sensors on our immune cells. They're designed to recognize common molecular patterns found on bacteria and viruses, PMPs, pathogen-associated molecular patterns. Binding of these PMPs to TLRs triggers an inflammatory response.
Okay, that makes sense for infection.
But here's the twist. Some of these same TLRs can also bind to molecules associated with atherosclerosis itself. like oxidized LDL.
Seriously. So the same receptor that detects bugs also detects oxidized cholesterol.
Some of them, yes, like TLR4. So the infection might prime the system, and then factors like oxidized LDL can keep activating those inflammatory pathways via the same receptors. It's a way infection could synergize with other risk factors.
So even if the infection itself isn't, directly building the plaque brick by brick, it might be activating or amplifying the inflammatory pathways that do contribute to it.
Exactly right. And this leads into something called the pathogen burden hypothesis.
Pathogen burden. What's that?
The theory here is that it might not be about one specific single infection causing heart disease. Instead, it could be the cumulative effect of being exposed to a greater number of different pathogens over your lifetime.
Like the total number of different bugs you've encountered.
Kind of, yeah. The idea is that this increased overall exposure, this higher burden, could lead to a state of chronic, low-grade systemic inflammation that just simmers along in the background and promotes the gradual development of atherosclerosis over many years.
Is there any evidence in humans to back up this pathogen burden idea?
Yes, there is some supportive evidence. Some epidemiological studies have looked at antibodies in people's blood, indicating past exposure to a range of different common pathogens, viruses, bacteria,
Yeah.
and they found that individuals who had antibodies to a larger number of different pathogens tended to have a higher risk of developing coronary artery disease. It suggests that the cumulative burden might indeed play a role.
Interesting. Okay, let's talk specifics then.
Right.
What about viruses? Herpes simplex viruses, HSV1 and 2, cold sores, genital herpes.
Mm-hmm.
Have they been consistently linked to atherosclerosis?
Well, the findings linking HSV infection to atherosclerosis have been somewhat mixed, not entirely consistent.
Okay.
There was a meta-analysis, which is where researchers combine the results from multiple individual studies to get a bigger picture, and that meta-analysis did show a statistically significant association between having evidence of past infection with either HSV1 or HSV2 and the presence of atherosclerotic disease.
So there's a link shown there.
Yes, but, and this is important, there was considerable variability or heterogeneity among the studies that went into that analysis, meaning the studies differed quite a bit in the types of people they looked at, how they defined exposure to the virus, like just antibodies or active infection, how they defined atherosclerotic disease outcomes, and ultimately in their results. Some showed a stronger link than others, some maybe didn't. This heterogeneity makes it harder to draw really firm, definitive conclusions just from that meta-analysis.
Got it. So suggestive, but maybe not slam dunk evidence. What about other common viruses like enteroviruses or hepatitis C or cytomegalovirus, CMV? Is the picture any clearer there?
For those viruses, the evidence linking them specifically to atherosclerosis is generally considered less consistent, maybe even weaker.
Okay.
Some studies have suggested a correlation between antibodies to enteroviruses and later cardiovascular events, but other studies haven't found that link. Similarly, the reported association between hepatitis C infection and heart disease has really varied across different studies.
And CMV.
For cytomegalovirus, a lot of the data comes from smaller studies. A meta-analysis looking at CMV suggested maybe only a modest association with coronary artery disease. So overall, for these particular viruses, the evidence just isn't as robust or consistent as, say, the ongoing investigation into some bacterial links.
Okay, let's turn to bacteria then. What about Helicobacter pylori? That's the bacterium famous for causing stomach ulcers, right?
Mm-hmm.
Has that also been implicated in heart disease?
Yes, H. pylori has definitely been investigated quite a bit for a potential role in atherosclerosis. Intriguingly, its DNA has actually been detected within coronary artery plaques in a significant proportion of patients in some studies.
Wow, the actual bacterial DNA inside the plaque.
Yes. Now, there have been some methodological concerns raised about potential contamination when you're looking for tiny amounts of DNA like that. But researchers have tried to use careful controls to address those issues, and the finding seems fairly consistent in some labs.
Okay, so the bacteria might be there. Is there epidemiological evidence linking H. pylori infection to heart problems?
Some studies have reported associations between having an H. pylori infection and increased risks of coronary artery disease, heart attack, and even stroke. There's also some suggestion that maybe the more virulent strains of H. pylori, the ones that are more aggressive, might pose a higher risk.
Any ideas how H. pylori might be contributing? What are the proposed mechanisms?
A couple of possibilities have been floated. H. pylori infection has been associated with lower levels of the beneficial HDL cholesterol in some people. It's also sometimes linked to higher levels of HbA1c, which is a marker for long-term blood sugar control, so it might influence traditional risk factors.
Okay. But is the evidence consistent here?
That's the issue. Similar to the viral data, the findings across large epidemiological studies have been pretty inconsistent. Several big, well-conducted studies have not found a strong link, particularly between certain H. pylori strains and death from cardiovascular causes.
Why the inconsistency, do you think?
It likely comes down to variations in how the studies were done. How did they define H. pylori exposure? Was it just antibodies or active infection? How did they define the cardiovascular outcomes? Were they looking at heart attacks, strokes, death, plaque buildup? And how well did they account for other factors, confounding factors, that could influence the results? It makes it hard to compare studies and get a clear answer.
Right, the classic challenges of epidemiology. Now, what about chlamydia pneumonia? That's another bacterium that was talked about a lot in relation to heart disease for a while.
Exactly. C. pneumonia, a common respiratory pathogen. Many initial studies, often smaller case control studies or early prospective studies, did suggest a potential link between having evidence of past C. pneumonia infection, like antibodies, and having atherosclerotic heart disease or stroke.
So there was early causative evidence.
Yes. However, when larger long-term prospective studies were done, following initially healthy individuals over many years, they generally have not consistently confirmed that association.
So the bigger studies didn't pan out.
Largely, no. And again, differences in how exposure to C. pneumonia was measured, how cardiovascular outcomes were defined, these probably contribute to the conflicting results we see in the literature.
Okay, now I definitely remember hearing about antibiotic trials back then. The idea was if C. pneumonia causes heart disease, maybe treating it with antibiotics could prevent heart attacks, right? What happened with those trials?
That was the logical next step, yes. Several large-scale clinical trials were conducted specifically to test this hypothesis. They took patients who already had established heart disease and gave them antibiotics known to be effective against C. pneumonia drugs like azithromycin or gadafloxacin, usually for a fairly extended period.
And the results? Did it help?
Generally, no. These major trials, some involving thousands of patients, have unfortunately not demonstrated a clear benefit in preventing secondary cardiovascular events, things like repeat heart attacks, strokes, or death in these patients.
So treating the C. pneumonia didn't seem to prevent more heart problems.
That was the main conclusion from the largest, most definitive trials, like the Wizard trial or the ACS trial. However, there is some other intriguing, although maybe less consistent, evidence from different studies.
Like what?
Some smaller studies or studies using different, perhaps broader spectrum antibiotic regimens or specifically the antibiotic roxithromycin have hinted at potential benefits. For example, maybe slowing down the progression of plaque buildup in the carotid arteries in the neck.
Okay.
And interestingly, in some of these studies where a potential benefit was seen, it didn't always seem to matter whether the patients actually had evidence of past C. pneumonia infection based on antibody tests.
Huh. So the antibiotic helped, but maybe not because it was killing C. pneumonia.
That's the implication. It raises the possibility that maybe other bacteria were involved, or perhaps the antibiotics were having some non-specific anti-inflammatory effects that were beneficial rather than targeting C. pneumonia specifically. It's still a bit of a puzzle.
Fascinating. Okay, one more area under infection. Oral pathogens. We often hear about the importance of good oral hygiene for overall health. Is there really a connection between, say, gum disease and heart disease?
There definitely seems to be a strong and pretty well-established association between periodontitis, which is severe gum disease, and clinical atherosclerosis.
An association. Okay, what does that mean in terms of risk?
It means that statistically, individuals who have periodontitis have been shown to have an increased risk of having a heart attack, a stroke, or peripheral artery disease compared to people with healthier gums. The link is quite consistent across many studies.
And have they found, like, mouth bacteria in heart plaques?
Yes, they have. Oral bacteria, the kinds that cause gum disease, have actually been identified within coronary artery plaques removed from patients.
Wow.
And the thinking now is shifting a bit. It might not be about just one or two specific bad oral bacteria. It might be more about the entire community of bacteria in the mouth, the oral microbiome.
It's a whole ecosystem again.
Yeah. The idea is that maybe having a greater number of different types of pathogenic oral bacteria could have a synergistic effect, working together to promote inflammation and potentially contribute to atherosclerosis development elsewhere in the body.
Okay, the association is strong. But is it a direct cause and effect relationship? Does gum disease actually cause heart disease? Or could it be that people with poor oral hygiene also have other unhealthy habits that increase heart risk?
That is the million-dollar question, and definitively proving direct causation remains a challenge. It's hard to untangle. We know that many risk factors for gum disease, like smoking, diabetes, poor diet, are also risk factors for heart disease.
Right, so maybe gum disease is just another marker of overall poor health.
That's certainly a possibility. The real test would be intervention trials. If you successfully treat the periodontitis, does it actually reduce the risk of future cardiovascular events?
And what have those trials shown?
They've yielded pretty mixed results, unfortunately. Some studies have shown improvements in markers of inflammation in the blood, like C-reactive protein, CRP, or IL-6 after successful periodontal treatment.
This sounds promising. Lower inflammation should be good.
It should be. But the larger, more rigorously designed trials that have looked specifically at hard clinical endpoints, actual heart attacks, strokes, deaths, have not consistently demonstrated a clear benefit from periodontal therapy.
So treating the gums didn't necessarily translate to fewer heart attacks.
Not in the biggest studies conducted so far. There was one major trial, the PAVE study, which specifically looked at this, and it did not show any reduction in cardiovascular events in the group that received intensive periodontal treatment compared to a control group.
Hmm.
Now, it's fair to say there were some limitations acknowledged in the design and execution of that study. For example, a significant number of people in the control group actually ended up receiving some periodontal care anyway, which could have diluted any potential difference between the groups.
Ah, okay. Makes it harder to see an effect.
Exactly. So the bottom line, while maintaining good oral hygiene is undoubtedly important for your overall health and the association with heart disease is strong, we don't yet have that definitive proof from large clinical trials that treating periodontitis will directly translate into preventing major cardiovascular events down the line. More research is still needed there.
Okay, this is a lot to consider. We've covered a lot of ground. Now let's move into a different category of these less common or less discussed risk factors.
Yeah.
Environmental toxins.
Right. Substances in our surroundings that we might not always connect directly with heart health, but maybe we should.
Exactly. What kind of things are we talking about here?
Well, one major group is persistent organic pollutants, or POPs. There's emerging evidence, and it's becoming quite strong, suggesting that certain POPs are associated with both atherosclerosis itself and also with type 2 diabetes.
And diabetes is a huge risk factor for heart disease.
A major risk factor, yes. So there could be direct effects on arteries and indirect effects via diabetes risk.
What are these POPs? What kind of chemicals?
It's a pretty diverse group. They include certain types of herbicides, insecticides, industrial chemicals like PCBs, polychlorinated biphenyls, and also dioxins. A particularly well-studied dioxin is TCDD.
TCDD. That sounds familiar.
It was a contaminant found in the Agent Orange herbicide, which was widely used during the Vietnam War.
Ah, Agent Orange.
Yeah.
Okay. And what makes these POPs persistent?
A key characteristic is that they are, well, persistent in the environment and in our bodies. They break down very slowly. They are also typically lipophilic.
Meaning they dissolve in fat.
Exactly. So they tend to accumulate in our body fat stores over time, and they can have very long biological half-lives, meaning it takes a long time for our bodies to get rid of them, if at all.
We've certainly heard a lot over the years about the potential health consequences for veterans who were exposed to Agent Orange. What's the specific link to cardiovascular disease that's been identified there?
The National Academy of Science, Engineering, and Medicine here in the U.S. has conducted ongoing systematic reviews of the scientific literature on Agent Orange exposure and health outcomes for many years.
And what have their reports concluded?
Their reports have consistently indicated an association between exposure to Agent Orange and several health conditions, including type 2 diabetes, ischemic heart disease, which is related to plaque buildup, hypertension, high blood pressure, and stroke. And importantly, the strength of the evidence supporting some of these associations has actually been upgraded over time as more research has become available.
So the link is becoming clearer.
It seems to be, yes.
Now, are these findings primarily relevant just to veterans with that specific Agent Orange exposure, or do they have broader implications for the rest of us regarding POPs in general?
No, the concerns definitely extend beyond just Agent Orange. More recent studies have also looked at broader exposure to various POPs in the general environment.
How do they measure that?
Sometimes through estimates based on where people live or work, other times by actually measuring the levels of different POPs in people's blood or fat tissue.
And what are these broader studies finding?
They have also generally supported links between higher exposure to various POPs and an increased risk of atherosclerotic disease, type 2 diabetes, stroke, and metabolic syndrome. And this is seen in both military veteran populations, beyond just Agent Orange, and in general civilian populations too.
But you mentioned some mixed results earlier.
Yes, it's important to acknowledge that. While the overall trend suggests a link, some studies have shown mixed or less conclusive results. It can sometimes depend on the specific POP being studied, the population group, how exposure was measured, and how outcomes were defined. But the weight of evidence is certainly pointing towards POPs being a concern for cardiovascular health.
Okay, so how might these POPs actually be exerting their harmful effects on our cardiovascular system? Do we know the mechanisms?
One key player seems to be a receptor inside our cells called the aryl hydrocarbon receptor, or ALHER.
Okay.
Many POPs, particularly dioxins and related compounds, are known to bind to and activate this ARR receptor.
And what happens when AHR gets activated?
It acts like a switch. It can trigger a whole cascade of downstream effects inside the cell, affecting gene expression. This can disrupt normal cell cycle regulation, promote inflammation, and interfere with various metabolic processes.
So AHR is a major pathway for POP toxicity.
It's considered a key mediator, yes. But there's also evidence for AHR-independent pathways. Some POPs might exert toxic effects through other mechanisms, perhaps by directly generating oxidative stress or by interfering with other signaling pathways that increase the production of inflammatory molecules.
Okay. And didn't you mention a connection back to TMAO earlier?
Yes. Crucially, some POPs have been shown to increase the activity or upregulate the liver enzyme FMO3.
The enzyme that makes TMAO from TMA.
Exactly. So this suggests a potentially really significant and concerning synergy. Your exposure to certain environmental pollutants could ramp up the enzyme that converts a dietary metabolite into the protherogenic molecule TMAO.
Wow. So diet and environmental toxins could be working together through this FMO3 pathway to worsen heart disease risk.
That's the hypothesis, and it's a very concerning possibility that needs more investigation.
That really is a troubling thought, that our food and our environment could interact in that way. Now, you also mentioned heavy metals as another category of environmental contaminants. Which ones are we talking about here?
Yes, besides the organic pollutants, certain heavy metals like arsenic, cadmium, and mercury, which can contaminate our environment through various industrial processes, mining, agriculture, and even natural geological sources, are another area of growing concern for cardiovascular health.
Okay, let's take them one by one. Let's start with arsenic. Where does exposure usually come from?
The most common route for chronic arsenic exposure for large populations globally is through contaminated drinking water. It can leach into groundwater from natural deposits or from industrial or agricultural runoff.
And what's the latest understanding of its impact on heart health?
Well, even at levels that are currently considered relatively low, according to regulatory standards set by organizations like the World Health Organization or the U.S. Environmental Protection Agency.
Which is what level?
The current recommended limit in drinking water is generally below 10 micrograms per liter.
Okay.
But even below that level, or certainly at levels slightly above it, chronic exposure to arsenic in drinking water has been pretty consistently linked to an increased risk of developing several serious conditions.
Like?
Type 2 diabetes, hypertension, coronary artery disease, stroke, and also peripheral artery disease, PAD. And some more recent research is even suggesting that there might still be an increased risk of cardiovascular mortality, dying from heart problems, even at arsenic levels below that 10 microgram per liter threshold.
So even the current safe levels might not be entirely safe.
That's what some of the emerging evidence suggests, yes. It's an ongoing area of research and regulatory discussion.
Okay, what about the next one? Cadmium. How are people typically exposed to cadmium?
Cadmium exposure can happen through a couple of main routes. One is through the diet. It can accumulate in certain foods, particularly shellfish, organ meats, and some plants grown in contaminated soil. The other major route is through inhalation.
Inhalation. From
Primarily from cigarette smoke, tobacco plants tend to accumulate cadmium from the soil, and also from industrial emissions, like from smelting or battery manufacturing.
I see. And what about its impact on heart health? You mentioned smoking complicates things.
Yes, Initially, because smoking is such a strong risk factor for heart disease and a major source of cadmium exposure, it was challenging to disentangle the independent effects of cadmium itself.
Right.
But more recent studies, carefully controlling for smoking history, have now shown pretty clearly that cadmium exposure, independent of smoking, is associated with a higher incidence of heart attack, stroke, coronary artery disease, and PD.
So cadmium itself seems to be a risk factor.
It appears so. There's also some evidence suggesting cadmium might play a role in the actual progression and stability of atherosclerotic plaques, maybe making them grow faster or become more unstable. Some research has linked higher cadmium levels to a greater density of macrophages within plaques.
Any idea how cadmium might be doing this?
Potential mechanisms include cadmium disrupting the normal function of the endothelial cells lining our blood vessels, perhaps making them more permeable or inflammatory. It's also known to increase the production of damaging free radicals, leading to oxidative stress. And interestingly, some research hints that these harmful cardiovascular effects of cadmium might be more pronounced in women compared to men, although the reasons for that aren't fully clear yet.
Interesting potential sex difference there. Okay, and the third heavy metal you mentioned was mercury. We often hear about mercury in fish, right? Especially certain types of seafood.
Yes, mercury is another significant concern. Mercury is released into the environment primarily from burning fossil fuels, especially coal, and also from industrial waste. In aquatic environments, lakes, rivers, oceans, bacteria can convert this inorganic mercury into a much more toxic organic form called methylmercury.
And that's what gets into fish.
Exactly. Methylmercury then works its way up the aquatic food chain. Plankton absorb it, small fish eat the plankton, bigger fish eat the smaller fish, and so on.
Yeah.
So it tends to bioaccumulate, reaching the highest concentrations in large predatory fish that are high up on the food chain, like shark, swordfish, king mackerel, some types of tuna.
Right, and this presents a dilemma, doesn't it? Because fish, especially oily fish, are also a primary source of those beneficial omega-3 fatty acids that are supposed to be good for your heart.
It's a very complex situation, absolutely. We're told to eat fish for the omega-3s, but we're also warned about mercury. How do you balance that?
So what does the research say about mercury exposure and heart disease risk?
The findings have been somewhat inconsistent, actually. There was a large, well-known prospective study conducted in Finland, for example. They measured mercury levels in people's hair, which is a good marker of long-term exposure.
Yeah.
And they found a strong association between higher hair mercury levels and an increased risk of having a heart attack or dying from coronary artery disease. Perhaps even more concerning, they suggested that the high mercury levels might have actually blunted or counteracted some of the expected protective effects of the omega-3 fatty acids consumed from fish.
So the mercury might negate the benefit of the fish oil.
That was the suggestion from that particular study, yes. However,
Oh.
it's really important to note that not all studies have consistently replicated this finding. Other large cohort studies have looked at mercury exposure and cardiovascular risk and haven't found such a clear detrimental effect, or maybe found no effect at typical exposure levels.
So the jury might still be out on mercury's impact relative to omega-3 benefits.
It seems the interplay is complex and maybe depends on the specific population, the levels of exposure, the type of fish consumed, and other dietary factors. It highlights the need for more research to fully understand this balance between the risks of mercury and the benefits of fish consumption.
Okay, one last environmental factor. We really can't overlook air pollution, can we? Especially for those of us living or working in urban areas, it feels like a constant exposure. What's the connection between air pollution and atherosclerosis?
Absolutely. Air pollution is a major environmental health concern with growing links to cardiovascular disease. Exposure comes mainly from sources like vehicle emissions, cars, trucks, buses, and industrial activities, power plants, etc.
And what's actually in polluted air that's harmful?
It's a really complex mixture. It includes gases like ozone and nitrogen oxides, but also tiny particles, particulate matter, often abbreviated as PM. These particles come in different sizes, with the smallest ones, ultrafine particles, being of particular concern because they can penetrate deep into the lungs and even enter the bloodstream. Plus, there are volatile organic compounds, aldehydes, and lots of free radicals mixed in.
Okay, so it's nasty stuff. Has it been definitively linked to atherosclerosis?
Well, establishing a direct, slam-dunk causal link between long-term air pollution exposure and the development of atherosclerosis itself has been challenging, mostly because it's difficult to accurately measure an individual's cumulative exposure over many years and to perfectly account for all the other factors that influence heart disease risk.
The confounding factors again.
Exactly. However, despite those challenges, a really significant and growing body of research does suggest a strong association between air pollution and various cardiovascular problems.
Such as,
Multiple studies have linked chronic exposure to common air pollutants with an increased risk of developing type 2 diabetes. And interestingly, that association sometimes appears stronger in women and in non-smokers.
okay, so it impacts diabetes risk. What about heart disease more directly?
Yes. Increased exposure to air pollution, particularly fine particulate matter, PM 2.5, has been consistently associated with higher levels of inflammatory markers in the blood, like that high sensitivity C-reactive protein, hsCRP, we talked about earlier.
So it causes inflammation.
It seems to trigger systemic inflammation, yes. And beyond just markers, studies have also linked higher air pollution exposure to a greater prevalence of clinically diagnosed coronary artery disease, stroke, ischemic heart disease in general, and even heart failure.
So it seems pretty strongly linked to bad cardiovascular outcomes, even if proving it causes the initial plaque is tricky.
That's a fair summary. The associations with events and risk factors are quite strong.
Do we have ideas about how breathing in polluted air might be negatively affecting our cardiovascular system at a biological level? What are the mechanisms?
Yes, several potential mechanisms have been identified and are being actively researched. As we just mentioned, triggering systemic inflammation seems key. Exposure to air pollution can also contribute to the development of insulin resistance, making your body less responsive to insulin, which is linked to diabetes and heart disease.
Hmm.
It can also activate the innate immune system, potentially through those toll-like receptors again, similar to infection. The high load of free radicals and oxidants in polluted air can induce significant oxidative stress throughout the body.
Damaging cells.
Yes, and potentially leading to mitochondrial dysfunction problems with the energy factories inside our cells. This oxidative stress and mitochondrial dysfunction are thought to contribute significantly to the development and progression of atherosclerosis at a cellular level.
And those tiny particles you mentioned.
Right, the ultrafine particles are small enough to actually cross from the lungs into the bloodstream. Once in circulation, they can directly interact with the endothelial cells lining our blood vessels, potentially causing direct damage, promoting inflammation right there at the artery wall, and contributing to endothelial dysfunction, which is an early step in atherosclerosis.
Wow. It really paints a picture of air pollution having multiple ways to harm our cardiovascular system.
It really does. Systemic inflammation, insulin resistance, oxidative stress, direct particle effects. It's likely a combination of these pathways contributing to the increased risk we observe.
So, as we kind of reach the end of this deep dive, it's incredibly clear that our understanding of atherosclerosis is, well, it's constantly evolving, isn't it?
Yeah.
It just keeps revealing more and more layers of complexity.
Absolutely. It's a continuously unfolding story.
It's definitely far more than just those classic risk factors, cholesterol and blood pressure, that we hear about most often.
Much more.
The intricate behavior of the plaques themselves, the surprisingly dynamic roles of the cells within them, changing jobs.
The shape-shifting VSMCs.
And the complexities of lipid metabolism beyond just LDL and HDL, like LPA. It all contributes to this much richer, much more nuanced picture.
And perhaps even more significantly, as we've really dug into today, there are these potential contributions from several less commonly considered factors, things that might be playing a bigger role than we used to think.
Right, like that intricate and very personalized relationship between our diet and the, trillions of microorganisms living in our gut.
The whole TMAO story and beyond, yes.
And the potential cumulative impact of our body's pathogen burden from past infections.
Still an area needing more clarity, but plausible.
And then our often unseen, maybe underestimated exposure to this wide array of environmental toxins, POPs, heavy metals, air pollution present in our air, our water, our food.
Things we're exposed to daily, often without realizing it.
It's almost overwhelming, isn't it, to think about all these different pieces, these different factors that could be interacting and playing a role in our long-term cardiovascular health.
It is indeed a complex picture, no doubt about it. But I think this deeper understanding, this appreciation of the complexity also brings a sense of hope, in a way.
How so?
Well, as we continue to unravel these intricate connections, as research clarifies these pathways, it opens up really exciting possibilities. Possibilities for developing new, maybe more targeted therapies in the future.
Right, like therapies targeting LPA or specific VSMC behaviors, or maybe even the microbiome.
Exactly. And also, it informs the potential for implementing more effective preventative strategies. Not just individual lifestyle changes, but maybe broader public health measures too, aimed at reducing exposure to some of these environmental factors, for instance.
That's a great point. It's not just about individual action, but potentially societal changes too.
Potentially, yes. Better understanding leads to better tools, both for treatment and prevention.
So, for you, our listener, as you've taken this journey with us through these less commonly discussed factors, maybe the final thought is this. Considering things like your diet's interaction with your microbiome, your history of infections, your potential environmental exposures, what aspects of your own health and your surrounding environment might be worth taking a closer look at, or at least being more aware of when you think about your long-term cardiovascular well-being?
It really makes you appreciate the profound interconnectedness, doesn't it, between our lifestyle, our internal biology, and the world around us.
It really does. A lot to think about.